do the recording again. Okay. So hi, everybody. It's a great pleasure to have Dr. Sharmin Ghaznavi from Mass General Hospital in Boston speaking to us today. Sharmin is the Associate Director and the Scientific Director of Cognitive Neuroscience at the Center for Neuroscience of Psychedelics at Massachusetts General Hospital. Very groundbreaking center in my mind and very promising. She's leading the center's first studies on the effects of psilocybin on cognitive processes in treatment-resistant depression. Dr. Ghaznavi received her undergraduate degrees in biology, brain cognitive and cognitive sciences, and philosophy from MIT. Very admirable. She obtained her medical degree, master's degree in philosophy, and PhD in neuroscience from Yale University. Dr. Ghaznavi completed a residency in adult psychiatry at the Mass General and McLean Psychiatry Residency Program. That's all in Boston and Belmont. Her research interests are the neurocorrelates of cognitive, affective, and social processing in psychedelic illness. Dr. Ghaznavi is very accomplished. She's been the recipient of the DuPont, Warren, and Livingstone Psychiatric Research Fellowships from Harvard Medical School and the 2012 NARSAD Young Investigator Award from the Brain and Behavior Research Foundations. I feel that we're very lucky to have you here. I'm very happy that we insisted on having you here. So the floor is yours. And,
okay. Moshe, thank you for the invitation. And as I was telling him before we started, this is my first time presenting to neuroscientists, largely because we haven't had data to present yet. But so I'm excited to hear what neuroscientists are thinking about this. I've largely been presenting to clinical, faculty and so on. So I want to begin with a little bit of clinical context. So I'm sure most of you have seen headlines like this one from the New York Times of a revolution in the works, and this one about how psychedelics like psilocybin will shake up psychiatry. And what I have to sort of convey today is that while psychedelics may prove to be extraordinary tools in treating mental illness, and I think, in terms of what we can learn from it about how the brain works and trying to understand how psychedelics work, I think talk of revolutions, might be a little hyperbolic. And so we'll sort of look at what the data actually shows at this time. So first of all, what do we mean by a psychedelic? So the term psychedelic was first coined by the British psychiatrist Humphrey Osmond, and it's derived from the Greek words psyche, which means soul or mind, and delos, which means manifest or visible. And so sometimes you'll hear psychedelics referred to as mind-manifesting drugs. In the 1950s, Osmond carried out research using mescaline and LSD and is known especially for his studies on LSD for alcohol dependence. He's also known for introducing the author Aldous Huxley to mescaline, who famously wrote The Doors of Perception and a lesser-known Heaven and Hell. And in fact, the band The Doors takes their name from his book, The Doors of Perception. So there are three major classes of psychedelics. They're the classic, and I should say there's some controversy about what's considered a psychedelic and how these are grouped, but this is sort of a basic one that most people agree on. So they're the classic psychedelics, also sometimes referred to as entheogens, because they've been used to produce non-ordinary states of consciousness for religious or spiritual purposes in different populations around the world. They're primarily agonists of the serotonin 5-HT2A receptor and include LSD, psilocybin, mescaline, which is the substance found in peyote, and then dimethyltryptamine, or also known as DMT, which is the major ingredient of the ayahuasca brew. Then there are the empathogens, which MDMA belongs to, so named because they induce feelings of empathy and relatedness. MDMA also acts on 5-HT2A receptors, but it's much weaker than the classic psychedelics. In addition, it binds to a number of other receptors, including adrenergic receptors, monoamine transporters, and prevents reuptake. Then there are the dissociatives, which include ketamine, ibogaine, and salve or salvinorin A, which is not pictured here. And the thing about these compounds is, aside from ketamine and salvinorin A is not scheduled. All of these compounds are classified as scheduled drugs in the United States. I'm not sure what the designation is in Israel, but it means that they cannot be legally accessed or prescribed except in the context of a research study for therapeutic purposes. So a lot of people ask about Oregon and Colorado, but they're not actually approved for therapeutic use there, or psilocybin is not approved for therapeutic use there, but more for exploratory use or recreational use, has been decriminalized. So what I'd like to do is go through a few of the clinical studies just to present for context and to understand why people are so excited about this. And also so that, you can think about how do we understand the neuroscience to sort of help improve clinical applications. So the first study that I'm going to go over with you is one that Moshe was beginning to talk about when I first joined. And it's probably the largest study done with psilocybin. And it was a randomized controlled double-blind trial of a single dose of psilocybin synthetic with psychological support in 233 patients with treatment-resistant depression. Now, psychological support is important to note here because most of the trials that I'll cover with you are trials of psychedelic-assisted psychotherapy. And so they often involve a significant therapeutic component or therapy component. Often there are three phases of it. There's preparation, which is when, sessions meant to sort of provide some psychoeducation, especially for patients who may be psychedelic naive, but also to help build alliance and to prepare for distress should it arise during the administration or dosing session. And then during the administration session, the therapy can differ depending on the particular protocol. So for instance, with most psilocybin trials to date, it's largely supportive and patients focus on their internal experience. With MDMA, it's a little bit more interactive and more directed. And then there's another component, the final component of therapy, which is often referred to as integration. And it happens after the dosing administration. And this is the opportunity for patients to describe their experience, understand it, and talk about how they make meaning of it, both from the experience itself and as they go about the world again. So in this particular trial, following a medication washout period, patients received 3 one-hour preparation therapy sessions. Then participants had a single 8-hour session in which they received either a 10 milligram or 25 milligram dose with 2 assisting psychotherapists. And the comparator arm received a 1 milligram dose. So one point about psychedelic studies is that it's very hard to blind for them because of the subjective and phenomenological effects of the compounds. And so sometimes researchers get around this by giving a lower dose of the psychedelic, or they'll still continue with a placebo such as niacin or in some cases mannitol. Here's a picture of a patient during a psilocybin dosing session. So you'll notice that they have headphones on. Often there's a playlist of music that they're listening to that has been somewhat curated to follow the experience of the compound that most patients have or participants have. And then also they often wear blindfolds. They don't have to, or not blindfolds, but shades. They don't have to, but many of them choose to, and they sort of go internally in the experience. They may get up and move and so on, but for the most part with psilocybin, they're focused on their internal experience, except when some distress arises. So in this study, they had an 8-hour single session, single dose, and then there were 2 integration meetings following the dosing session. And the integration sessions are really participant-directed or self-directed inquiry. The primary endpoint for this study was the change in MADRS score from baseline. And as you can see, there were effects even the first day, but the most significant difference was at 3 weeks for the 25 milligram group. And so they just launched their phase 3 trial with a dosing of 25 milligrams. So we'll see how that data comes in. Some of you may have heard of the New England Journal of Medicine article about the study comparing psilocybin to escitalopram. And that study was a double-blind, randomized control trial involving patients with long-standing moderate to severe major depressive disorder. And they were comparing psilocybin to escitalopram over a 6-week period. So patients were assigned in a 1-to-1 ratio to receive 2 separate doses. So in this case, this is 2 doses of 25 milligrams of psilocybin 3 weeks apart, plus 6 weeks of a daily placebo if they were in the psilocybin group, or 2 separate doses of 1 milligram of psilocybin 3 weeks apart, plus 6 weeks of daily oral escitalopram. And in the escitalopram group, they had a 10 milligram dosing with an option to escalate to 20 milligrams at week 3. All the patients received psychological support similar to the Goodwin trial. And what was interesting was that their primary endpoint was the QIDS-SR, and they did not find a significant difference between the psilocybin and escitalopram groups. Now, some of you may have seen that the authors did provide a number of secondary outcomes, which suggested that psilocybin outperformed escitalopram, but those were not corrected for multiple comparisons and don't show significance when there is a correction for multiple comparisons. And this study is important to note because I'll be presenting data later on from some of the neuroimaging trials that actually took this sample and conducted scans on them. More recently, Franz Wallenreiter's group in Switzerland examined the effects of a single moderate dose of psilocybin. So this is about 15 milligrams compared to 25 or 30 milligrams is done in some of the Hopkins studies versus placebo. In this case, they used mannitol in 52 individuals with MDD. The trial was double-blind, randomized, placebo-controlled with mannitol, and involved 2 preparation sessions and 3 integration sessions, 2 days, 8 days, and 14 days after dosing. They found a significant decrease in the symptom severity on the MADRS and BDI in the psilocybin condition compared to the placebo condition. A major controversy or issue up for debate in psychedelics literature is whether or not the subjective or the phenomenological experience under psychedelics is responsible for the therapeutic effect or necessary for the therapeutic effect. And so Wallenreiter's group actually took a look at this, and they assessed it using the global intensity score of the altered states of consciousness, which is a measure that's commonly used in psychedelic studies to sort of measure the subjective effects of the psychedelic compounds. And what they found was that there was no significant difference in global intensity scores assessed by the altered states of consciousness between responders and non-responders at 14 days. So the intensity of the psilocybin-induced subjective effects did not correlate with the reduction in depressive symptomatology at 2 weeks post-treatment. And this is in contrast to earlier studies, which seem to suggest that the subjective experience matters. And now I'd like to briefly go over the results of the first of 2 phase 3 trials for MDMA-assisted therapy for PTSD. So the trial was a randomized, double-blind, placebo-controlled, multi-site trial to test the efficacy and safety of MDMA in 90 patients with pretty severe PTSD. These were folks with CAP scores greater than 35. There were 3 preparatory sessions, 90 minutes each, with 2 therapists. Again, the therapy was largely to, build alliance and develop skills should distress arise during the dosing sessions. With the MDMA trial, there's a total of 3 8-hour administration sessions, as well as 3 hours of integration therapy following each dose. In the first session, an initial dose of 80 milligrams was followed by a supplemental half dose of 40 milligrams, about an hour and a half to 2.5 hours after the first dose. In the second and third experimental sessions, there's an initial dose of 120 milligrams, followed by a supplemental half dose of 60 milligrams, and patients could always decline the supplemental dose. The picture here shows a reenactment of an MDMA administration. Again, here you'll notice they're wearing headphones. They have shades on. That said, in MDMA therapy, patients are much more talkative and there's more active therapy going on than in psilocybin. They looked at CAPS total severity score from baseline to following after the third session. And they found a significant reduction in the CAPS-5 total score from baseline to the final time point. And interestingly, it drops off significantly even after 2 doses. So they are beginning to explore whether or not 2 administration doses might take the place of 3, just given the cost of being able to provide this treatment. Then this bar graph shows the percentage of people who are responders. That's defined by a greater than 10-point decrease in CAPS, those who no longer met CAPS diagnostic criteria, and those who met CAPS criteria for remission. 67% of the participants in the MDMA group no longer met diagnostic criteria for PTSD after 3 sessions, compared with 32% of those in the placebo group. Additionally, 33% of participants in the MDMA group met criteria for remission after 3 sessions, compared with only 5% in the placebo group. Recently, Bogenschultz and colleagues published the results of a double-blind randomized clinical trial in 95 patients with alcohol use disorder. So participants in this study received 12 weeks of a manualized psychotherapy, which combined components of motivation enhancement and CBT. And participants received either psilocybin or diphenhydramine during 2 day-long medication sessions at weeks 4 and weeks 8 in the therapy process. And they found a significant decrease in the percentage of heavy drinking days for the psilocybin group compared to the diphenhydramine group. They also found a significant decrease in drinks per day. Additionally, they found a decrease in percent of drinking days, but this did not reach significance. And most importantly, those in the psilocybin group had a 2-level decrease, 2 levels of decrease in the WHO risk level compared to those in the diphenhydramine group. So even if they didn't abstain, they've made a significant difference in their risk profile. So now I'd like to switch to discussing more of the neuroscience. So one of the reasons psychedelics are thought to be catalytic in conjunction with psychotherapy is research, especially preclinical research, showing that psychedelics increase the brain's capacity for change or neuroplasticity. As I'm sure all of you are aware, all healing, whether through medication or therapy or neuromodulation, is realized by changes in the brain. And what allows this change, right, of course, is neuroplasticity, our brain's capacity to change. Again, I want to emphasize that this is based on preclinical work in animals and usually rodents. And so, there's the classic study showing that psychedelics promote structural plasticity. Here you see that cortical neurons of rats that have been exposed to serotonergic compounds or classic psychedelics such as DMT and LSD show increased dendritic spines. They also there's also increased synaptogenesis. And what's important is psychedelics not only promote structural plasticity, they also have been shown to promote functional plasticity. So here we see that DMT increases both spontaneous excitatory postsynaptic current, frequency, and amplitude. In 2021, the study came out showing increased dendritic spine density and spine width in mouse cortical neurons, even just 1 day following psilocybin administration. In that same study, Shao and colleagues demonstrated not only that there's structural and functional change, but that it correlates with behavioral change. So they trained mice on a behavioral task, resulting in learned helplessness. So the mice stopped trying to escape even when there's the possibility, right? But notably, even among mice, there are those that are more resilient and there are those that are more susceptible. And they did distinguish between these populations. And what they found was that with psilocybin administration, among mice more susceptible to learned helplessness, those who received psilocybin were more likely to actually try when there was the possibility to escape after they had received the psilocybin. Going one step further, it turns out that psychedelics might even reopen critical learning periods. So as a little bit of background, and some of you may already know this, mice show a preference for an environment in which they are with other mice. And this is called social place preference. Interestingly, around 90 days after birth, they lose this social place preference. And so Goldolin and her colleagues explored whether or not it was possible to open this with MDMA. And what they found was that when mice were administered MDMA, they again show a social place preference, thus reopening the critical period for social reward learning. And hot off the presses just last week, she'd been presenting this data for a while, but just a couple of weeks ago, another paper came out showing that this is the case for other psychedelics as well, including psilocybin, LSD, ketamine, and ibogaine. So if it's above the wave, that means that it's reopened the critical learning period for social preference. What's interesting is that she also showed that the duration of during which the critical learning period is reopened is proportional to the duration of the acute subjective effects of these drugs in humans. So those with the longest subjective effects in humans, like psilocybin, MDMA, LSD, and ibogaine, showed longer opening of that critical period. More recently, Beta Moghadam's group examined the effects of psilocybin on cognitive flexibility in rats using an operant set shifting task that some of you may be familiar with. So rats were required to respond to one of the ports according to either a light rule, where the lit port was rewarded, or a side rule. So the port on a specific side was rewarded regardless of which one was lit. If and when rats reached a criterion of 10 consecutive correct responses, the rule was changed. Each session lasted until criterion was reached on 4 rule sets or until 45 minutes elapsed. And what they did was they administered psilocybin to see how that would affect the ability to shift sets. And what they found was that psilocybin did in fact improve performance and average trials to criterion across all sets in the baseline period. And here they're looking at response time, and this is even though they were slower because they were under the effects of the psilocybin. Interestingly, they also assessed the effects of psilocybin on reversal learning. So they now asked the rats to be able to reverse associations with an appetitive versus an aversive conditioned stimulus. And this time they found that psilocybin did not have any effect on reversal learning. So it didn't facilitate changing prior programmed responses. And this is something worth bearing in mind when we move to our discussion of human neuroimaging studies, and especially a lot of discussion about how one of the things that may be happening with psychedelics is that people are changing prior associations with psychedelics. Another reason that psychedelics are believed to help in psychiatric conditions is because of their anti-inflammatory properties. You know, there's a lot of research going on about how inflammation may play a role in psychiatric conditions. And there's research using DOI, which is a 5-HT2A agonist, and administering it in a model of acute asthma in mice. So it turns out that if mice are exposed to ovalbumin, they actually have an inflamed response that mimics asthma. And so what they did is they administered DOI to mice to see how they would then respond to exposure to ovalbumin. And what they found was that the animals that had been treated with DOI exposure prior to being exposed to ovalbumin actually had normal airway morphology, whereas the untreated animals showed the peribronchial inflammation and mucous cell infiltration. And what's interesting is that 5-HT2A receptor activation represses the expression of inflammatory mediators in multiple tissues. So psychedelics are also being explored for their potential role in preventing inflammatory conditions or treating inflammatory conditions. And another interesting thing is that the in vitro data with regards to potency and efficacy of this anti-inflammatory effect shows that it seems to be seems to not correlate with canonical signaling pathways, so that it might be possible to achieve it without necessarily having the subjective effects. And that's important as people are considering synthesizing new compounds. Now what I'd like to do is switch to human neuroimaging studies and discuss some of the mechanisms of action that have been proposed to underlie therapeutic properties of psychedelic studies. An important thing to keep in mind is that some of these studies are done looking at acute effects, either during or right after the psychedelic compound is administered. But there isn't a lot of follow-up. So there isn't information across multiple time points and following people further out. So that's something to keep in mind. So one of the first studies was done by Franz Ballwinder's group, and they examined the effects of psilocybin on regional cerebral glucose metabolism in 10 healthy volunteers with PET and F18, FDG. And this was following a 15 or 20 milligram dose of psilocybin. They did it weight-based, but it's supposed to be high dose. And what they found were increases in cerebral metabolic rate of glucose in the prefrontal cortex, in the anterior cingulate, temporomedial cortex, and putamen. And this led them to propose what's called the thalamic filter model. And so this model suggests that given that the thalamus plays a key role in controlling or gating information to the cortex, the core effects of psychedelics are thought to result from deficits in gating activity. This in turn is thought to result in increased activity in the prefrontal cortex and other cortical regions, and mediated through the disintegration of information processing within the cortico-striato-thalamo-cortical feedback loops. In a follow-up study from the same group, they examined resting state activity, connectivity in these feedback loops after acute LSD and after pretreatment with ketanserin. So ketanserin blocks 5-HT2A activity in a double-blind randomized placebo-controlled study in 25 healthy participants. The resting state scans were required twice, 75 minutes and 300 minutes after treatment administration. They used dynamic causal modeling, and they found that LSD increased effective connectivity from the thalamus to the posterior cingulate cortex, dependent on 5-HT2A activation. They also found that LSD decreased effective connectivity from the ventral striatum to the thalamus and the PCC, and the thalamus to the temporal parietal cortex. Increased effective connectivity from the posterior cingulate to the ventral striatum, and reduced self-inhibition of the PCC, irrespective of 5-HT2A activation. So overall, that there was decreased gating of activity between thalamus and cortical areas. So Carhart, Harris, and colleagues out of Imperial College at the time examined arterial spin labeling, perfusion, and bold signal before and after intravenous infusions of placebo and psilocybin in 15 healthy volunteers. In contrast to the earlier PET study by Vollenweider's group, they actually found decreases in cerebral blood flow and bold signal, and these were maximal in the thalamus and the anterior and posterior cingulate cortex. In fact, the most consistent finding was decreased activity in the anterior cingulate and medial PFC, and the magnitude of this decrease predicted the intensity of the subjective effects that the volunteers reported. Psilocybin caused a significant decrease in the coupling between the medial prefrontal cortex and the PCC. And this led Carhart, Harris, and colleagues to hypothesize that, so whereas Franz Vollenweider was saying that there was an increase in sort of bottom-up activity, and it's driven from the bottom up through gating, Carhart, Harris, and colleagues were sort of presenting this more like top-down processing theory, and that what happens, and what they propose is that psychedelics, like psilocybin, exert their effects by decrease in top-down processing and an increase in entropy. And this is, behind his like snow globe effect metaphor, that somehow priors are loosened like the snow in a globe that has been shaken and resettles. In a later study, Carhart, Harris examined cerebral blood flow and resting state functional connectivity before and 1 week after treatment with psilocybin for treatment-resistant depression in 16 patients. They again found decreases in cerebral blood flow, this time in the temporal cortex, including the amygdala. And what's more, the decrease in cerebral blood flow correlated with reduced depressive symptoms. So here shown as a greater reduction in amygdala cerebral blood flow directly related to a greater reduction in the QIDS score. However, when they examined resting state functional connectivity in the default mode network, they actually found increased functional connectivity in the default mode network. In fact, increased ventromedial PFC to bilateral inferior lateral parietal cortex resting state functional connectivity was predictive of treatment response at 5 weeks. And so this led Carhart, Harris, and colleagues to propose a reset mechanism by which they think initially default mode network integrity is decreased acutely or normal, and then increased or normalized post-acutely. Of note, while this hypothesis explains the disparate findings between these two studies, it hasn't actually been tested because it requires scanning acutely and post-acutely, which has not been done. Last year saw the following headlines in the news regarding a much talked about article that was published in Nature Medicine. And the headline specifically suggested that psilocybin rewires the brain and frees it up from patterns of rumination. And I'd like to cover some of the data just that this is based on. So the study that these headlines are based on examined resting state fMRI activity at baseline and 3 weeks after treatment with psilocybin. And it's the same population sample from the New England Journal of Medicine article. And most notably, there was no significant interaction between the treatment arm and scanning session on network modularity. So they didn't actually have an interaction that suggests that there's a difference between psilocybin and escitalopram. And despite that, they go on to look at what happens in the psilocybin arm. And what they report is that, that consistent with their previous hypothesis, which, is not clear because they should expect a reset mechanism, significant reductions in default mode network recruitment, that there were significant reductions, and that there was increased between network integration between the default mode network and between the default mode network and salience network 1 day after psilocybin therapy. But recall that previously they showed increased network connectivity, right? So, one, there was no interaction to follow up with, and this was discussed in various editorials, but also this was not consistent with prior findings. So it's interesting because they still, claim that there's a lot of compelling evidence in the paper suggesting that modularity is related to the antidepressant effects of psilocybin. Now, with regards to the claims about freeing people up from patterns of rumination, they examined the same data set to look at the effects of psilocybin versus escitalopram on rumination. They also looked at thought suppression from 1 week before to 6 weeks after treatment. As some of you may know, rumination refers to excessive self-focus on one's symptoms and causes of one's current distress without actually engaging in active problem solving. So this is the vicious cycle of, I shouldn't have done this, and I brought this on myself, and, I'm so miserable. Thought suppression, of course, refers to trying to actively keep certain things out of mind. So, I have up here the rumination scale because what's important to realize is what they actually examined is a trait variable because they don't have a state measure of rumination. And this trait measure is basically the tendency to engage in rumination. Same was true for thought suppression. So they claimed that they measured whether people are ruminating or suppressing thought, but that requires measuring a state variable. And it's not a trivial issue, right? So for example, for the measure here, you'll see that they ask people, what do you generally do in response to feeling sad, down, or depressed, right? So if one is a responder to psilocybin or escitalopram, and you're not feeling very depressed over the previous few weeks, how can you talk about how you generally would respond at this point, looking at it 6 weeks out? And also importantly, this still doesn't capture whether or not they would actually go on to still have this tendency to ruminate, because most trait measures are measured not just a few weeks out, that you're looking for reliability over time, right? And that's the very definition of a trait. And so, yes, could traits be changed? Possibly. Could personality measures be changed? Yes, but they need to be followed out much further than 6 weeks. And so one could hypothesize that psilocybin changes trait rumination or other trait variables, but proving that requires retest reliability, which has to be done over and over for a longer period of time. But I'll still go through the data with you right now. So with regards to the claims about freeing people up from patterns of rumination, they examined the same data to look at the effects of so they looked at psilocybin versus escitalopram. Again, it's on the same group that was before. And remember that there was no difference between the two groups, right, on the quiz SR. But what they found here, when they ran in ANCOVA, they found a two-way interaction significant for time and condition. So there was a time by condition interaction. So the condition being, psilocybin or escitalopram. So those who had received psilocybin supposedly showed a decrease in trait rumination. But what's interesting is that when they do a three-way, they found a time by response interaction, and not a time by response by condition. So there was a decrease in trait rumination, not just in the psilocybin group, but also in the escitalopram group. Another mechanism that has been proposed is that psilocybin leads to increased flexibility. So Das and colleagues examined the effects of psilocybin therapy on cognitive flexibility, which they measured by looking at perseverative errors on a set shifting task outside the scanner. And they also wanted to look at neural flexibility and brain regions supporting cognitive flexibility, and that have been implicated in acute psilocybin effects, like the anterior cingulate and posterior cingulate. And they did this in 24 patients with depression. They found that psilocybin therapy increased cognitive flexibility for at least 4 weeks post-treatment. They also found that static functional activity generally decreased across the brain, and that dynamic functional connectivity generally increased across the brain. However, when they examined neural flexibility, which they defined as dynamic functional connectivity between the anterior and posterior cingulate, they found that at baseline dynamic functional connectivity between the anterior and posterior cingulate predicted improvements in cognitive flexibility, but that greater increases in dynamic functional connectivity were associated with less improvement in cognitive flexibility after psilocybin therapy. So the cognitive flexibility and neural flexibility, as they defined it, didn't quite map. Yet another hypothesis is that the mechanism of action of psychedelics is claustrum-mediated cortical dysregulations is another bottom-up. And so, like as many of you know, the claustrum highly expresses 5-HT2A receptors and also provides glutamatergic inputs to all areas of the cerebral cortex. So Fred Barrett and colleagues examined whole brain resting state activity in 15 healthy participants 100 minutes after psilocybin versus placebo. And they found that psilocybin decreased the amplitude of low-frequency fluctuations and variance in the claustrum bilaterally. They also showed that network integrity, which they defined as within network functional connectivity and modularity, is associated with claustrum connectivity in networks including the default mode network and the frontoparietal task network. And here they show it again in plot format. And then a final hypothesis that has been put forward is that there is decreased negative affect with the administration of psychedelics, especially MDMA, that makes it possible to process difficult material or change post-administration perception of negative material. So Carter Harris and colleagues tested this by investigating the effects of MDMA on recollection of favorite and worst autobiographical memories in 19 participants with previous experience of MDMA. And they found that MDMA augmented activations to favorite memories bilaterally in the fusiform gyrus and the somatosensory cortex, and attenuated activations to worst memories in the left anterior temporal cortex. And there's been one study that has assessed the effects of psilocybin versus placebo on amygdala reactivity to negative stimuli in 25 healthy participants. And they found that amygdala reactivity to negative and neutral stimuli was lower after psilocybin administration than after placebo administration. Additionally, the psilocybin-induced attenuation of right amygdala activity response to negative stimuli was related to the psilocybin-induced increase in positive mood state. These were healthy participants, so it was more like, could they increase positive mood? So a couple of things that are really important. To date, there have been about 51, maybe a few more since in the past couple months, human neuroimaging articles of the effects of psychedelics. Of those, 42 articles are on resting state fMRI. And importantly, and this review covers it nicely, they vary in data processing and analysis methodology. So there isn't a lot of consistency. But what's really important is that 2 data sets are the foundation for half of the published literature. So 52% or 22 of the 42 studies is derived from 2 original data sets. And those 2 original data sets are the only 2 studies ever done with IV administration, whereas many of these studies that I've covered are oral administration for clinical purposes. And participants in both studies were exclusively healthy individuals and had previous experience with psychedelics. And so I wanted to end by talking about sort of, a challenge for psychedelics is, all the data is resting state data. And as we know, resting state networks differ in important ways from networks underlying cognitive function, right? Regions activated during rest are not always activated during cognitive tasks, and the converse is also true. And the same brain region can be involved in multiple cognitive functions, and its pattern of functional connectivity can differ across cognitive functions. And so I wanted to share this graphic. Some of you may have seen it from Erin Finn in a recent TICS paper. And, she talks about how the first wave of functional neuroimaging consisted largely of investigators using these highly controlled tasks, right, to isolate specific cognitive processes. And while this approach, could generate and localize small ripples in brain activity, ultimately it ignored a large fraction of variance that arises from, task unrelated, seemingly spontaneous signals. And then we saw the second wave of neuroimaging where investigators dove headlong into the depths, right, to characterize more of the resting state activity. And, in the clinical realm, this is very much done, and also because it's easier to do than tasks. But because we have very little to sort of anchor our observations to, it's difficult to interpret these signals and understand how it actually relates to cognition. And so the third wave of research in cognitive neuroscience, I think, especially for psychedelics, will be for us to partially, come back to the surface and, as she describes, and actually regain some experimental control so that we can, while, appreciating the importance of, spontaneous generated activity, to be able to really understand what is going on. And finally, I began this talk with a lot of discussion about the headlines of revolutions shaking up psychiatry. And again, while I think there's real promise in doing work in this area for psychedelics to help some of our patients significantly, these are early days yet, and talk of revolution seems a little hyperbolic. I'm reminded of a line from Thomas Kuhn's Structure of Scientific Revolutions. Though the world does not change with the change of paradigm, the scientist afterward works in a different world. And I don't know that we've gotten there yet. It's still early days for the neuroscience of psychedelics as well. But arguably, the neuroscience to date does not provide a revolution, evidence of a revolution in sight just yet. But it's definitely exciting times ahead. I'd just like to thank my team. So this is the scientific leadership of the Center for the Neuroscience of Psychedelics, and below is the team assisting me with our first studies in psilocybin. Thank you. I'm happy to take questions.
Thank you very much, Sharmin. It's been fantastic. So actually, I have too many questions. Maybe I should open the stage for others first before I steal all your attention. So if you have a question, just unmute yourself and go ahead. Okay, so I'll ask one. So you showed a few possible accounts for the effect of psilocybin on depression, and none of them seem satisfactory to you, and and I think also to me. The question is, what's your intuition? Without, feel free to speculate. What do you think if I just put you on the spot and ask you, how do you think psilocybin improves symptoms of depression? What do you think will be the most likely?
I think that it is. I think that it is. I think the preclinical data is pretty compelling, and I think that's the most rigorous science to date, right? And showing these opening, this increased capacity for neuroplasticity and opening critical periods. And so I think that in the recent, I didn't cover this, but there are recent data showing that the binding to tract B, is so high, which are, found on BDNF receptors. So the plasticity here, I think, is significant. And so I think it's opportunity. it's interesting that all of this has been done with psychedelic assisted psychotherapy. And I think there's a reason. It's like opening up the window and being able to make changes. Do I think it happens as radically and quickly as people report? I'm not sure. Because a lot of these clinical trials, for instance, the very early clinical trials, especially open label trials, were people with prior experience and expectancy going in. So, you know, are we going to find that single dose studies continue to make that impact? No. Is there a lot of room for thinking about what we expose people to following opening these, this opportunity? I think, yeah, there's a lot of opportunity there. There's also a lot of risk, right? So just as a reminder, plasticity is neutral, right? So bad things can happen too, right? People can become more susceptible. So we have to be mindful of that as well, especially because, many of our patients are going to be going back into potentially difficult circumstances in their life, right? And we need to be thinking about that as well. So, in terms of how I think there is this increased capacity to change how we think about things and how we view the world. But I think that we have not proven yet what actually changes in terms of how people think and feel differently. We have narrative reports, but that's different from understanding what's changing in the brain.
Excellent. Okay, so it's the plasticity. A slight technical question in your very early slides when you show the chemical composition of the different psychedelics. So it struck me how the psilocybin and DMT seem to highly overlap, right? I mean, I'm not an expert in this, but there seem to be like high overlap. But I know from friends that DMT has a very short effect, where psilocybin can last a few hours. Do you have any explanation for the difference in duration?
No, I don't know. I mean, in terms of like the actual chemistry, that's outside my wheelhouse as to why one lasts longer than the other. And often DMT is not taken alone. So the ayahuasca brew actually also includes an MAOI inhibitor, right? So it prolongs the duration of DMT. So in naturalistic settings, people are not just experiencing a few seconds of DMT or a few minutes of DMT. That said, there is a company that's done an inhaled formulation of DMT, interestingly, without any therapy, granted in only 8 patients with TRD, and found that they achieve remission at 14 days. No long-term follow-up yet, and just that preliminary dosing study. So in terms of why the duration is greater, I don't know. You would have to ask somebody that has a little bit more knowledge in terms of the chemistry. Like, what about that? Yeah.
I was happy to see his picture there. Yeah. And last question from me. So there's been a lot of noise, talking, not noise, but a lot of discussion about the effect of placebo in depression and how SSRIs, the majority of its effect can be accounted for by placebo. I'm not sure I'm completely convinced, but it does seem like when you show the psilocybin versus different types of SSRIs, they seem to deviate. And I wonder what do you think is a component of, placebo seems to me even less likely in psilocybin, but what are your thoughts about, the Compass, for example, and all these other trials? What do you think, to what extent do you think placebo played a role?
Well, I don't, so the comparator trial is it wasn't properly powered, or, and like, for instance, if people needed a 20 milligram dose of escitalopram, we know to follow them out 6 weeks beyond, right? And they got 3 weeks beyond. So it wasn't properly designed to really, to look at efficacy, right? And what it showed actually was that there was no difference, right, in terms of the primary endpoint. And so, but I think, in terms of whether there's a placebo, I think that there's a huge expectancy bias. I mean, it's really hard to blind for psilocybin, right? And what their published results don't show is that it was like about 33% or something that actually had a response. So not everyone does, right? So that's the other thing. It's not a cure-all. It's not a panacea. Not everybody responds. And that's really important too, because people come to it thinking this is their last hope. And the reality is some people don't sometimes respond to psychedelics, but then do for other treatments if they've received adequate treatment. So I think, the idea of placebo here, and I'm not sure that I would call it placebo so much as expectancy. There's also a lot of therapy going into these treatment protocols, right? And like you saw with the MDMA-assisted psychotherapy, the response, even just from the therapy, is pretty significant, right? I mean, the fact that maybe not for remission, but for a decrease in CAPS score, it was pretty significant. Some of these people have not received adequate treatment before. And so there's that component too, but if they're going into this study, they're highly motivated, it's highly controlled. But this idea of whether one would have more placebo than the other, if anything, it's more that expectancy is going to be greater, but there are expectancy biases with SSRI studies too. There's functional unblinding with most drug studies because of side effects or whatever, people kind of know.
Yeah. Okay, great. Very informative.
I wanted to ask. So just wanted to introduce myself. I'm a new faculty here, and I worked at UCSF with Joshua Woolley and with Robin Card-Harris. I was also a neuroscientist and facilitator for these studies. I worked with Usona and Philomen, and so I've been in the room. I saw how it happens. I wanted, first of all, to thank you for a wonderful lecture, wonderful talk about this. It's all very important. I think people should know about the difficulties surrounding this type of study and this type of research. And I wanted to ask for your opinion as a clinician and a neuroscientist in this field about microdosing and also about non-psychedelic psychedelics. So since both of them are being researched and are being kind of in the making, I think, in trying to understand if they can help psychiatric disorders. So if you have anything, to share about that.
So, microdosing is generally done by people at home, right? So, the studies to date have been sort of naturalistic survey studies. And, even they show that microdosing doesn't seem to help really. So people report, all of Silicon Valley will tell you that, microdosing really helped me and it made a big difference, but at least the research doesn't seem to suggest that it makes a difference. Now, some people might say, well, then why not, what's the harm in microdosing, right? And I think that this sort of doesn't take into account the fact that some of these compounds, and psychedelics are not monolithic, right? Like for instance, the cardiac risk for ibogaine is much higher than it is with psilocybin, but these do have effects on cardiac valves. And what is repeated exposure? We don't know what those risks are. And it doesn't mean don't proceed, but we don't know what they are. And so we should be advising caution because people can't make a clear risk-benefit analysis, right? And so in the presence of studies that show no benefit, I'm not sure that the potential risks are worth engaging in. With regards to non-psychedelic psychedelics, right? Here's the thing. I think about it as a clinician. My expertise is in bipolar disorder, and those patients are excluded from these trials. So in case people aren't aware, if you've had an experience of psychosis, or mania, but also if there's a first-degree relative, in some cases, second-degree relative with a history of psychosis or mania, you're excluded from these trials. So we don't have great safety data on it. But additionally, it means that these treatment options and the plasticity effects may not be accessible to you or may not be available to those patients. And so I'm excited for the possibility of non-psychedelic psychedelics. And sometimes they're referred to as psychoplastogens, right? Because it's trying to capitalize on the neuroplastic potential without necessarily having the subjective effects that present such massive risk for patients that are otherwise excluded. And, I'm excited for the potential of building that up because, I'm not wedded to the subjective effects. I understand the benefits for some people and the profound experiences people can have. But if some of my patients can benefit from the plasticity effects without the subjective effects that would be so dangerous to them, I'm all for it and welcome the possibility of it. And I don't think that devalues the subjective experience. It's just not appropriate for everybody to have that experience. And I think that should be, the benefits, some of the benefits should still be an option for people.
Thanks.
Sure.
Okay, any more questions?
Can I just ask if you can, like...
Go for it,
Yeah. I would then just like you to expand a bit more about what you said about the subjective experience that some of those patients go through. And I would really, I've been exposed to some of the research done by private companies about ibogaine, and the use they make of it to less with the depression, more with the addiction problems. And they claim to have some successes with creating the effect you were describing about non-psychedelic experience, while still creating a plasticity effect that was sufficient for pretty good withdrawal. But do you think it's possible to achieve that kind of plasticity without having any subjective experience, in your experience?
So, I didn't present this data, but the recent data showing 1,000 times greater affinity for track B. I mean, that is how the plasticity, compared to traditional antidepressants. And that is completely aside from the 5-HT2A activity that underlies the psychedelic effects or the subjective effects, right? So it seems completely possible, right? So if you design a compound that is, and the company you're talking, the compound you're talking about with regards to ibogaine, I think it's called it's a mouthful. It's like turbinatholog or something. It's like similar to, it's supposed to have some of the plasticity effects as that the subjective effects. Of course, they've only used it in animals thus far, or at least in terms of published stuff, right? I think that it may, it's an empirical question if it's possible, but it seems like it might be possible based on how we understand mechanism. Whether we call them non-psychedelic psychedelics, I don't know. I think psychoplastin probably makes more sense because the psychedelic effects are the subjective effects. Some people describe it as awe. Some people describe, there are perceptual changes. With the dissociatives, it's more of a sense of unreal, they don't start to have, such perceptual disturbances. So ibogaine and, ketamine, ketamine people just feel a little bit out of it and dissociated, but it's not like psilocybin or MDMA.
Okay. Well, thank you very much, Sharmin. It's been a real pleasure.
Thank you for having me.
Informative. We'll be in touch.
All right. Take care. Bye-bye.
Bye-bye.